So the first time that I was teaching A&P2 and I got to this section, I kind of thought, yeah, well, okay, blood vessels, capillary stuff, that's going to be easy and not actually very interesting. That's actually not true. So today's lecture is really important because it's the first time that we're going to look at materials moving across cell linings and generally across membranes in this class. And in this class, stuff moving between organs or out of the blood and going somewhere else, or from somewhere else into the blood, moving through cell linings or membranes, that keeps on happening. In the stomach, in the intestines, in the kidneys, in the lungs, in the capillaries. So this lecture is super important. And it's probably good that I'm recording it because you can go back and watch it later if you find yourself like completely confused by what's going on in the kidneys. It's related to this. So we're going to learn a lot about blood vessels, and they're more than just straws or tubes. They're actually much cooler than that. Most blood vessels, except for capillaries, those are an exception, most of the other blood vessels have 3 layers. And the layers of these organs are called tunica something. So you need to associate tunica in your memory with anatomy of blood vessels. So tunic, like coat, clothing. We have tunica interna, internal, that makes sense. That's the endothelium. It is an epithelial lining, for those of you that do not like those. This is an epithelial lining. Generally acts as an epithelium. Then we have tunica media, that's the middle, and that's a muscle layer. The thickness of the muscle layer is different in different blood vessels. That one changes quite a bit. And then the outer layer, or tunica externa, external, that's mostly connective tissue and wrappings that hold the whole blood vessel together. So, tunica interna, media externa. We've got epithelial lining, muscle lining, connective tissue. So basically all the parts except nervous tissue in the blood vessels. Again, it's the size of the muscle layer that changes the most. And what we're looking at here, you should label which one is the artery and which one is the vein. And there's 2 clues in the picture. Which one's the artery?
The one without the valve.
The one without the valve. Yeah, the valve is a clue. Valves are in veins sometimes. And also the artery has much more muscle to it. Arteries have thicker muscular walls. So this is the artery, this is the vein. And for a given diameter of blood vessel, if it's an artery, more of the diameter is taken up by muscle. If it's a vein, more of the diameter is taken up by internal space in the lumen. You know that word lumen? That word keeps coming around in this class too. So the lumen is the inside of an organ. The empty space, the lumen. So that's the width of a lumen of a vein of that diameter. That's the width of an artery lumen for a given diameter. And if you look at them down here, we'll look at this in lab next week. If you see a blood vessel and it's got really a lot of space and not much muscle layer, that's a vein. This one's got a lot more muscle thickness to it, less internal space, that's an artery. And they tend to run along next to each other.
Can you repeat the definition of lumen?
Yeah, the definition of lumen, this is a lame definition of lumen, but it's the empty space inside of an organ. And there's usually a reason for it to be empty because stuff is supposed to pass through it. Yeah, lumen, super useful word. the space inside. This question, the answer to this is because they need it. Veins need bigger lumens. And this is kind of jumping ahead, but why do veins need bigger lumens?
Because they have the most blood content, don't they?
Because they have the most blood content. They have the most blood content because they have the biggest lumens. It's kind of like a reverse thing. This was not obvious to me when I started teaching, but they need bigger lumens because they need to have less resistance to blood flow. Veins have lower pressure in them. We're going to see that 3 or 4 more times next week, but veins, the blood in the veins is under less pressure than the blood in arteries. So if the lumen is more wide open, it's less resistance against blood flowing back to the heart. We'll come back to that again later. So those pictures that we just looked at, those would be, veins and arteries when their muscle layer is in a relaxed state. So sort of neutral, relaxed state. Either of those, either a vein or an artery, can be signaled by the autonomic nervous system. There's that again. That keeps coming up all through the semester as well. To either cause vasoconstriction or vasodilation. I feel like I say those 2 words every lecture. You should memorize their definitions. Vasoconstriction, the blood vessels are clamping to a smaller diameter, so their diameter decreases. That would be like vasoconstriction, smaller diameter. Or when they relax and are not constricted, that's vasodilation. So they're opening up, the lumens become bigger, the muscle is less constricted. Diameter increases. So memorize these 2 terms, their meanings if you haven't already. Keeps on being important in various places in the body. Also, this is showing you, how big an arteriole could be when the muscles are relaxed versus when they're contracting. And I really like these pictures because it's showing red blood cells passing through. So it's giving you an idea of how big is a red blood cell, how big is an arteriole, and this is not the smallest. That's not the smallest artery yet. So another thing that you should memorize, and you know these words probably already, but this is the systemic circulation, another way of thinking about it. So when blood leaves the heart through the aorta, it's going to take this general path through blood vessels from biggest to smallest. So you might want to draw an arrow over top of that row. That's going from biggest to smallest arteries. And when you get down to the very smallest arteries, they then merge into capillaries. Capillaries aren't classified as arteries or veins. They're in between. They're very tiny. They're all tiny. And then, when you get onto the vein side, the blood is coming from capillaries into venules, into veins, into the vena cava. So this row is going from smallest on this side to largest on that side. The bottom row is arranged from smallest to largest. And when we say systemic, when we talk about systemic circulation, this is the systemic circulation. Except, different body parts, they have names for the arteries in that region, names for the veins in that region, but that's the order that blood is going through. Does it kind of make sense? That's details of the systemic circulation.
So, the first one was with what, biggest to smallest? Or what?
Yeah. So in this list of arteries, the aorta is the biggest artery that you have that's coming out of the heart. That's as big as it gets. And then it'll start branching into arteries, and the arteries branch into arterioles. Coming up in lab on your blood vessel list, we call that part of the heart. I mean, it's connected to the heart. It is a blood vessel. The names that you need to know on the artery side, they're at that level of size. We don't name arterioles because there's too many. I know you think there's already too many names of arteries, but those are just the big ones. And then the same on the vein side. Vena cava is attached to the heart. That's the biggest vein that you can have, veins. The ones on your memory list are vein size. Venules are too small to learn the names of. So here's a somewhat of a depiction of how that literally would look. So what you're looking at here, it's helpfully colored, red for arteries, blue for veins. That's helpful. Using the usual colors here. And we're looking right now at, this is an arteriole. It's a very small artery. This is a venule, a very small vein, and the capillaries are in between, connecting them. So what we've got is blood coming into arterioles. It's very oxygen rich. It's then moving through capillaries, and it's hard to see on here, but it's going from red on this side, and the further it goes, the bluer it gets. So in here, if you've got this picture, make a circle or a bracket and write on there, it's in the capillary beds where tissues are doing gas exchange. That's where the gas exchange is happening. Capillary beds. So what's going on here is blood comes into this region, oxygen leaves the bloodstream overall and goes into the tissues. What's moving the other way? Carbon dioxide is coming out of the tissues into the blood. So the blood that comes in this way is high in oxygen coming into the capillary bed. Blood that's leaving the capillary bed is lower in oxygen and higher than CO2. And we call that gas exchange. So during gas exchange, the blood somewhat swaps O2 for CO2. That's oversimplified, but that's a way to start thinking about it. Swapping with the tissues. It would be like if one truck came to your house and it brought you groceries and it also took away trash. That would be great. We don't really have that system yet. Someone could start a business for that. It would save gas. So the next thing I want you to think about is, okay, we're going from big arteries down to arterioles, down to capillaries. How really small the tubes are, the blood vessels are that we're talking about. They're going to get really small. And what we're looking at here, this is an arteriole on this part of the branch. And down here, branching off, is a capillary. And they're made out of individual endothelial cells. They're seriously tiny. So, this capillary, the whole diameter of it is made from about 3 cells wrapping around the circumference of it. Tiny, tiny, tiny. But the muscle bands, when you get down into the arterial layer, they're not all along the length. They're just like hair bands tying off a braid or something like that. They're just little bands. They're not a continuous wrapping. down at this level. So can an arteriole change diameter? As long as there are muscle bands to constrict or relax, that means that the blood vessel can change diameter. So an arteriole can get bigger or smaller. Can a capillary get bigger or smaller? It is what it is. It doesn't have muscles that can change its size. It's just endothelium. Why do capillaries not want, so to speak, why do they not need to have muscle layers? It's related to what they need to be doing.
Gas can exchange through.
So they're supposed to be doing gas exchange. That's in the capillary bed. And if you want good gas exchange, the gases need to move through thin spaces, so to speak. Thin spaces. Through thin spaces. So it's much easier for gases to move through this part of an arteriole than to try to wiggle through this muscle layer also. It's too thick. It's too thick. So if we think about, go back to our list of big and small. The layers of these, except for the very smallest arterioles, these don't do gas exchange, they're just blood movers. Gas exchange needs thin walls to move through. Has to be in the capillaries. Which also means that the capillaries have to have thin walls to do gas exchange. They don't need muscle layers, they need thinness to do their job. So part of how capillaries do their job of delivering nutrients and delivering oxygen, that's half their job. The other half of their job is to take away wastes and CO2. So they need to be delivering good stuff and taking away bad or pointless materials from the cells. They do that in two ways. Maybe I'll write this also over here. They're picking up wastes, they're delivering nutrients and oxygen. And whatever materials are moving in and out of the capillaries there's two ways that can happen. Some of the materials will actually be wiggling through gaps in the endothelium itself. So there are little slits, there are little gaps, there are actual physical spaces in the capillary walls that let some things go in and out. And this is just barely a wall. It's very puny. It's kind of leaky. So things can go in between the cells. If they fit. Not everything can fit through that space. It has to be small. Or there are some things that can go through the cells of the endothelium. They don't have to go through the slits. They actually whoosh through the cell itself. And it depends on the size of the thing and the nature of the substance as to which one it moves through. It might be both. It might be only through the slits. It might be mostly through the cell, or it might not be able to get out at all. It depends on the size. But the movement of things from the blood out to the tissue fluid is really important. Questions? Seem okay? What is this, do you think? This is not in your book. What's it look like?
Network.
It is a network of capillaries. And it's from a living organism that you've seen this organism a lot. This is a horse hoof. So this is, hoof shape down here. This is the capillary network in a horse hoof. And I think it's so cool. You hardly ever see the true configuration of capillaries because they're so small. Like, you can't dissect that out. So the way that these are done is they take a plasticizer and run it through the blood vessels, and that solidifies plastic in the shape of the capillary bed, and then they get rid of the rest of the tissue. Plastic remains. If you go to those human body exhibits, they have things like this, and it's cool. Plasticized capillary networks. But you get to see there are capillaries in there everywhere, which makes sense. Aren't you glad that you don't have to memorize anything but the major blood vessels? They go everywhere in your body. So because gas exchange happens in capillaries, capillaries need to go everywhere in the body, basically. You've got to have them all over your body. So if we have some cell in our toe, it can't be too far from a capillary because it's depending on the bloodstream to bring it oxygen, to take away its CO2, to bring it nutrients, to take away its wastes. You can't have cells that are just really far from the capillaries. Just kind of like in human civilization, you can't have people that are like so far away from roads, generally. You got to have the connections to the materials. So capillaries go everywhere. And
By caps, you mean capillary?
Yeah, I just didn't write the whole thing. Caps, capillaries. Caps means something different in soccer. It means how many games were you in, right? Rest of the day, caps or capillaries in here. So if this is a cell in my toe, it's got to be pretty near to a bloodstream. But it's not always touching a capillary. It's got to be near, but it's not touching. So in between there's fluid. And that can have two different names. What I just wrote, I know it's tiny, I wrote tissue fluid. Tissue fluid. So if you think about all the cells that are in the horse hoof, they're not all literally touching a capillary, but they're all near to one, and there's tissue fluid in between. And that's the part that maybe you haven't thought about before. So here's a capillary, there's blood going through it. All around it there's fluid. And within the fluid there are also cells. So the tissue fluid is in between cells. It's in between cells and blood vessels. It can be called tissue fluid. It can also be called interstitial. Slow down on that. Interstitial fluid. I'm making a mess of this word. Interstitial fluid. Or tissue fluid. And this is, if you think about blood vessels as roads, and cells like households, this is like when the mailman has to walk from the car up to your porch. It's not that far. It's not that far. There's a limit to how far they will do that. Maybe that they prefer to reach out and put the mail in the mailbox, but they might walk up a little bit to your house. That's like tissue fluid. So that sets up, we've got the blood vessel part, we've got the cells that they're serving, we've got the interstitial fluid. So here's another picture of a capillary bed or a capillary network. moving from arteries, branching off into arterioles, which can branch off into the capillary bed. And this is where cells are getting their gas exchange and nutrient exchange, in here. Makes sense because it's moving from totally red to totally blue. So if you imagine cell, 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 and tissue fluid in here. None of them are too far away from a capillary. If your body needs to, it can actually constrict some of the arterioles that supply this and shut it off if it needs to. So that it's like saying these cells can wait a little bit because there are other cells elsewhere that need the blood materials more. And you feel this every day. For example, if you go and eat a lot of pizza and like a lot of carbs, and then you just kind of sit down and you feel sleepy, guess what? The capillary beds in your digestive system are preferred at that time over capillary beds in your brain. It's not just, a coincidence that, oh, I'm sleepy. Oh, I'm sleepy. There's literally more blood going to your digestive system at that time and less to your brain. If you're running away from a lion, or you are, in the third period of your wrestling time, I don't know if girls have the 3, guys do, and you're really doing a lot of exercise, the capillary beds in your digestive system are shut down. And somewhat, the capillary beds in your brain might be shut down, slightly, because your muscles need so much oxygen right now. If you've ever tried to play a sport that requires thinking and you're not very fit, this was me back in the day, like, I can run or I can think about the play, but I cannot do both of them at the same time. Once you get really fit, you can, but your muscles will be preferred over the brain. And you felt shunting before. Capillary shunting is what this is called. You felt that before. It makes sense. These cells can wait till later. There's other ones that need more attention right now. So you have a place on your notes to write examples. What would you write for examples? That should be where we are. What are examples of when capillary beds would open or shut?
You just named like 3 or 4.
Yeah, I did.
So those are some.
Yeah. I did just name 3 or 4. Though you might want to know some examples. Great, good. All right. Now this is probably the first time in the class when we get into really detailed physiology, and this is called capillary dynamics. So these next 5 slides, this next stuff that I'm going to say is super important. It will be an exam question on exam 2. And we're going to spend a lot of time on these next 4 slides. They're really important. So if you find this confusing, you'll be able to go back to the video and go to whatever time it is right now and watch it again. There's also a review video that relates to this that's already on Blackboard to talk about how the materials get in and out of capillaries. So anytime that you have a picture that you really need to understand in detail, you need to know what all the parts are. And what we're looking at here is this, which is slightly cut off. This is an arteriole on this side, and it's showing like half of it. This is a capillary, and you notice it's colored from red moving toward blue. It's like we sliced through a capillary. What would be over here on this side? There'd be a venule, not shown. The white stuff is tissue fluid. What's that thing? That's a cell, a bunch of cells, surrounded by tissue fluid. There's a lymphatic vessel right here, and we haven't talked about the lymphatic system yet, but this picture becomes important for that body system too. Down here on this end, you would have the same arrangement on this side, but it's not shown. And what this picture is about is fluids going out of the capillary and fluids coming back into the capillary. So this is really about fluid movements into and out of the capillary. The other important part of this picture are the slits between the capillary endothelium. Things can get in and out through those small slits. So what's going through the capillary generally right now? There is oxygen. What else? Blood. Generally blood. I don't know why that musical sound happened right now. There's blood going through. It's a liquid. It's got a liquid component and it's got solid components. So there's red blood cells, there's plasma, there's white blood cells, there's platelets. What else is in there? Nutrients, oxygen, sugar, all kinds of things. So we have liquid. There's liquid moving. And there are, again, two reasons that make liquid move in your body. This word is via, is what I'm trying to write. Via. There's liquid moving via two forces. Hydrostatic pressure. That one, like, we understand really well. It's like when you turn on a faucet and water comes out, you say, yeah, I've got water pressure. If the water is not coming out, I have no water pressure. This shower is awful because it has low water pressure. You're used to that concept. That's hydrostatic pressure. How much is the liquid pushing on stuff? Hydrostatic pressure. Oh, okay. Second thing is harder to get your mind around. It's osmotic pressure. Osmotic pressure. And this one depends on diffusion. So think back to when you talked about diffusion back in A&P 1 or back in high school. What is diffusion? Say that again.
I can just go, yeah.
High to low concentration?
Yeah.
Spreading out?
Yeah.
I like to think of it. Spreading out. So molecules will tend to spread out. That's a property of our universe. And they will try to move from where they're in high concentration to low concentration if they can. If they can. Sometimes they can, sometimes they can't. And to think about that concept, you have to know, okay, for the thing I'm talking about, can it go across or can it not? We'll come back to that in a minute. But when the blood first comes into the capillary, it's under pretty high hydrostatic pressure because the heart pushed it there. What part of the heart pushed it here? What part of the heart is responsible for getting this blood under pressure? It is the left ventricle. Because remember, this is the systemic circuit. The left ventricle pushed it here. Squeezed it through the aorta, through all the blood vessels. It's coming in under pressure. So when it arrives here in this kind of leaky capillary, it's under higher hydrostatic pressure, and the hydrostatic pressure is pushing out. It's like if you had normal hoses and then you had a soaker hose down here, it's got holes in it, liquid starts coming out. So water starts coming out these little holes under hydrostatic pressure. Does that make sense? Because these are leaky.
Yeah.
coming out the holes, under hydrostatic pressure. So I want to make over here a list of what can go through capillary slits. What can and what can't. I know this is off the screen, but off the video, but you can still hear it. So think about what's in the blood. Of the things that are in the blood, what can go through slits? What's small enough? Gases. O2 and CO2. For sure, they're tiny. What else could fit through there? What kind of energy? Glucose. Proteins, only if they're tiny. Only if they're tiny. Which means that if proteins are big, they can't fit through. What else could fit through? Amino acids. What else? There's lots and lots of things that could fit through. Electrolytes. Enzymes would fall under proteins, tiny and big. There's a really important one you don't have yet in this one. Water. I like it. Water. Water can go in and out. Water is small. So there's a molecular cutoff in a capillary slit between what stays in the bloodstream and what can leave. So we've got the general set of small stuff that can fit out through a capillary slit. What does that leave behind besides big proteins? What's stuck?
Red blood cells.
Red blood cells. What else? White blood cells. Platelets. Or formed elements here of the bloodstream. And big proteins. Within the class of big proteins, put albumin and globulins. This is one of those kind of minor points from the first unit. And sometimes if I ask a question on the exam about a minor point it's because it becomes important later. These are important now. So this stuff is staying in the bloodstream. It cannot leave the capillary. Small stuff can. This is so convenient. This is so convenient. Look what's in this small stuff. It's nutrients and oxygen. How convenient that they can get out here into the tissue fluid toward the cells that need them. Toward the cells that need them. Guess what? A lot of the wastes that these cells make, like CO2 and other wastes, they're also small enough to go into a capillary as well. So, at the very least, those small things can get out of the bloodstream and into the tissue fluid, closer to where the cells need them to be. Now, whether or not they get across the membrane right there, that's your active passive transport stuff. We'll talk about that another day. But that's great. All of these small, useful things can get through those little holes, get into the tissue fluid, close to where the cells need them. So this is filtration. This is filtration. We've got little stuff squeezing through holes under hydrostatic pressure, and filtration is putting nutrients into the tissue fluid. I think I need to change the slides. It's been on that forever. That's the easy part, I feel like. That's the part that makes sense and that we're sort of used to thinking about. That's the outward force. So the hydrostatic pressure, that's like water pressure. Filtering, pushing things out. This is the one we're going to talk about next, osmotic pressure. So osmotic pressure, I already have that on the slide, I don't need to write it. Osmotic pressure is the tendency or the ability to pull water across a membrane. That might be different from a book definition that you read, but I wish I could write on this screen. You can write on your picture, right over here in the tissue fluid, that after all this stuff comes out into the tissue fluid, that still on this side there is more water. There's more water here, generally less water in here. More water up here, less water down here. Write that on your slide. And then if you have a different color of pen, you don't have to, but if you do, you might want to write something else on those two sides. If there's more water here, that means that there's generally less solute here, less dissolved stuff on this side.
And that's tissue, where tissue and stuff.
In the
How
tissue
white
fluid.
stuff,
Yeah, in the tissue fluid. So this side of the membrane generally has more water and generally less solute, partly because these cells are sucking up nutrients all the time. So the nutrients don't hang around here very long, the cells gobble them. So on this side there's more water, there's less solute. On this side there's less water, there's more solute. So the osmotic pressure will make water go down its concentration gradient. And the picture is calling this inward force of osmotic pressure 24 millimeters of mercury. If I were you, I would write water moves down its concentration gradient next to that arrow, because that's really what that means. Water is going from where it's high to where it's low. That's the osmotic pressure. That's the second force. Water moves down its concentration gradient. That's what this orange arrow is showing.
And the yellow is solute.
The yellow is hydrostatic pressure. That's another important part of these figures. Like the yellow arrow that they're showing, that's hydrostatic pressure filtering out. The orange is water getting pulled back into the bloodstream because of solutes here. And then there's another arrow, one other color of arrow that they're showing. Is that on there? Okay. This arrow, which they call net outward pressure. Is it okay? I know this is heavy stuff for a Friday afternoon. You know what this is? I really like it, actually. These arrows have a mathematical relationship, and that's what they're showing down here. This arrow is the combination of forces that you would get from adding these two together. So you have a large outward force, the hydrostatic pressure, and they give you a number on that, 35. 35 millimeters of mercury, that's just a unit of pressure. That's a hydrostatic pressure. The osmotic pressure is directed inward with a value of 24, so it has an opposite sign of that. And if you subtract the small from the big, you get the net force, which is 11 out. That part is putting numbers on the concepts. Putting numbers on the concepts, that's how the exam, I'll ask you on the exam. I'll say the hydrostatic pressure is directed outward with a level of 42, and the inward osmotic pressure, the osmotic pressure is directed inward with a level of 16. What's the net pressure, and how is water moving in this area? Here's how you do that. draw a picture of it. This picture, you want to really study this picture. So we've got hydrostatic pressure pushing water out at that level, 35. We've got osmotic pressure pulling water into the capillary at that level, 24. Those are in opposite directions. Which force wins in that? The outward. The outward is the bigger force. So you take the difference between them, and that gives you the net, the overall force, which is outward at 11. The numbers make sense? How do you do the numbers? I'll have some practice problems next week for you. So that's the situation in the beginning of a capillary bed. There's a lot of force pushing outward because the blood comes in under hydrostatic pressure. And there's inward osmotic pressure pulling water back in for these solids. So make a note that these big things that can't get out of the blood, they are considered solutes that are always stuck in the bloodstream. They can't get out. They're too big. But water can move in either direction that it wants to, through the capillary slits. So when blood first comes into a capillary bed, overall, water is moving out from the capillary bed to the tissue fluid, and it also would be carrying, the small stuff with it. It's moving out. That's at the beginning of the capillary bed. So now we start to look at the other end. This part has not changed. So we were looking at this part before. This is the beginning of the capillary bed. All the numbers are the same. All the arrows are the same. We're just moving down a little bit now. So if we think about this as a soaker hose with holes in it, water will be pushing out a little bit harder when it first hits the holes than when it does later. Does that make sense?
Can you say that one more time?
Yeah. You know what I mean by soaker hose? You might not. Like, you all don't go buy hoses a lot, probably. A soaker hose is when you have a regular hose attached to the spigot and hopefully it doesn't leak. They make these hoses that you can put around your flowers or your vegetables that have holes in them on purpose so that you can connect that to the normal hose and it will slowly leak water in an efficient way without you having to go water stuff. They're great. I don't have any. I need to get some. That's what I mean by soaker hose. So if I ever talk about something like that and you just have no idea, don't be afraid. Like, what is that? What is a soaker hose? They have other names too. I don't remember what they are. But if we have a place now where there's water coming through some holes and it's under pressure, the first place it hits the holes, it's going to be coming out faster. Because the further it goes, the more the pressure dissipates. Does that help? How many people had not heard of a soaker hose? Kind of makes sense. It's not high up on your list of items that you need in your life right now, but they're really useful in under certain situations. So the more the blood is pushing this way, the pressure just kind of drops off. Pressure's dropping off. So there's less water pressure, there's less hydrostatic pressure going out down on this end. The hydrostatic pressure has gotten less. It's just kind of petering out. So down here on this end, it's 16. It's 16. Up here it was 35. And that's just because a lot of water has already leaked out. But there's still osmotic pressure. There's still big solute stuff in the blood that will pull water back in. And so this arrow that represents the value of the osmotic pressure, that hasn't changed. It stays the same. It's still 24. So now, we've got 16 pushing out, 24 pushing in. Which one wins now? Osmotic pressure is now more than the outward hydrostatic pressure. So when we look at the total pressure, the sum, it's a small inward arrow with a value of 8. What does all this mean? What does all this mean? Here's what it generally means. At the beginning of a capillary bed, fluid overall is moving out. It's leaking out. Imagine if that was the end of it. Your blood would not retain enough liquid in it. So at the beginning of a capillary bed, you've got fluid moving out. At the end of a capillary bed, you've got fluid moving in. Osmotic pressure pulls fluid back in. Pulls fluid back in.
So hydrostatic pressure gets rid of fluid, osmotic pressure brings it back.
Kind of.
Kind of.
Yeah. Yeah. The last thing I want you to write on this slide, and we will continue with this slide set on Monday, is make a, like a star or a circle or something and write, but not all of it. Not all of it. So lots of fluid leaves on this end. Most of it comes back in on this end, but not all of it. Not all of it. Kyler, if you would hit the